Hello viewers, the aim of this activity-based video is to demonstrate that carbon dioxide is released during the process of respiration. Let us see the experimental setup first. In this experimental setup, we have 3 glass tubes, 1 conical flask. It's an airtight experimental setup. In test tube A, we have taken potassium hydroxide solution. In test tube B, we have taken lime water. In conical flask, we have taken germinating pea seeds. And in test tube C, again lime water is present. Now this experimental setup is connected to a suction pump. So when we will switch on the suction pump, it will start sucking the air and the air will start coming to test tube A. From this, it will move to test tube B, and like that, it will move out through the suction pump. Now coming to the steps of demonstrating this experimental setup. Germinate the pea seeds by soaking them in water overnight. Place germinated pea seeds in the conical flask. Start the suction pump and let the air flow through the apparatus. As the air passes through test tube A, which is containing potassium hydroxide solution, this solution will absorb all the carbon dioxide gas present in the incoming air. So this air, which is now devoid of carbon dioxide gas, goes forward to test tube B. The air entering test tube B passes through lime water, but no change is noticed in the color of lime water. Had carbon dioxide gas been present in the air, lime water would have turned milky white. But there is no change in color of lime water. This confirms that there is no CO2 in the air that has entered in test tube B. This air, which is devoid of all carbon dioxide gas, leaves test tube B and enters the conical flask, which contains the germinated pea seeds. The germinated pea seeds utilize this air in carrying out respiration. Now, this air further moves from conical flask and enters test tube C, which contains lime water. As the air passes through the lime water in test tube C, the lime water turns milky white in color. So on the basis of these observations, we can conclude that carbon dioxide gas was being produced in the conical flask, which had germinated seeds in it. This gas moved from conical flask and entered into test tube C. So with the help of this experimental setup, we can prove that during respiration, carbon dioxide gas is released. Now coming to the precautions. First, this experimental setup should be completely airtight. Second, the germinating seeds should have moisture. Third, freshly prepared potassium hydroxide solution should be used. Fourth, the ends of the capillary tube should be completely dipped in the lime solution taken in test tubes B and C.